Hello, ladies and gentlemen, and welcome to our holiday lecture series in which we discuss all the reasons why we are thankful for DNA. Now, in our lectures, what we're going to do is look at the elegant design of DNA that was first elucidated by James Watson and Francis Crick, building on the ideas and research done by individuals over a number of decades here. We'll also look at how this hereditary information is replicated so that it can be passed on to the daughter cells that make up multicellular organisms, and the significance of all these concepts. The significance of DNA and its organization is that DNA plays a significant role in the reactions and structure of our body. So DNA sort of plays a significant role in influencing how our bodies are put together and how our bodies operate. Now here's a lovely picture, sort of a well-known image of Watson and Crick admiring their elegant explanation for the structure of this model. Now, early on, people weren't particularly familiar with the structure of DNA, but it came to be looked at a little bit more closely after the work of Thomas Hunt Morgan and his lab in which they studied the behavior of chromosomes in fruit flies. Now, from their studies, we knew that genes, these hereditary structures, are found on chromosomes, but the issue became, was this hereditary information stored in the DNA, or was it stored in the protein? Now, proteins were an early leader in this search for the structure of the hereditary molecule, because of the diversity of protein structures. There are so many different protein structures with very precise functions. So people thought this heterogeneity or these differences in proteins made them a more probable candidate for the molecules that store hereditary information. Now, in 1928, the work of Griffith actually helped begin to point the needle in the arrow, or the arrow in the direction of DNA as being the hereditary molecule, because in his work, Griffith was actually able to transform bacteria. He took virulent or harmful heat-killed bacteria and mixed them with these non-virulent or harmless bacteria that were living. And what happened is the non-harmful or the harmless living bacteria were converted or transformed into pathogenic bacteria after taking in material from the heat-killed pathogenic bacteria. He called this process transformation because these non-pathogenic bacteria were made pathogenic after taking in or assimilating foreign DNA. Now, to sort of visualize this, we can look at these bacteria that are covered with this capsule. These are the virulent bacteria. This capsule helps make them resistant to the immune systems of the mouse. So the pathogenic bacteria have these capsules, and if mice receive the injections, they develop pneumonia and they die. Now, there are strains of the bacterium that do not produce these capsules, so they are non-virulent or non-harmful. If mice are injected with those non-virulent bacterial cells, they're fine. Now, if Griffith heated up the bacteria in order to destroy these capsules and denature the proteins in the bacterium, again, the mice are unaffected. But if he mixed the bacterial cells, these virulent bacteria that had been heat-killed, with living non-virulent bacteria, these non-virulent or non-pathogenic bacteria were transformed into pathogenic bacteria and caused the mice to divide. Furthermore, when they looked at the bacterial cells that were taken from these mice that had died, the bacterial cells had been again transformed into the pathogenic strain. So he knew that information was being passed on from these pathogenic cells that had been killed to these non-pathogenic cells. And because of the heat treating, he had assumed that the proteins would have been denatured, so that DNA was a likely candidate for the substance that was passed on to the non-virulent bacteria. Now, that did not settle the debate by any means. So it was over 20 years later that Hershey and Chase were able to conduct an experiment that sort of laid to rest the controversy related to the genetic material of organisms. Now, a virus is a fascinating structure because it is basically a protein body, or what's called a protein coat, that wraps around some genetic material or DNA. Now, we know today that viruses inject their DNA into a host cell, and then they, in one way or another, cause the host to produce viral proteins. Now, at the time of Hershey and Chase, they didn't know what was being injected from the bacterium into the host, but they knew something must be injected because viruses require a host in order to divide. Now, the viruses they chose are structures called bacteriophages. This is an important term. Please make sure you're familiar with the concept of a phage. But bacteriophages are viruses or phages that require a bacterial host. Now, in their experiments, what they were able to do is go through and tag DNA and proteins in a virus or in these particular viruses or phages, and then track the radioactive material. And in doing so, their conclusion was that what was actually injected into the bacterial host was the DNA. The genetic information was DNA. Now, to visualize what they did was grow these bacterial cells, or I'm sorry, these viruses in radioactive sulfur. Now, sulfur makes up the disulfide bridges found in proteins. It helps proteins keep their three-dimensional structure. So they knew that if proteins are what were injected into bacteria, then the bacterial cells that they were able to isolate would glow from the radiating sulfur. Now, they also grew a second strain that included viruses labeled with radioactive phosphorus. Now, DNA has its phosphate group, so the phosphorus found in DNA would then be injected into the bacterial host if it was the DNA that is actually inserted. So they cultured or grew the viruses and let them infect the host bacteria. Then they separated the viruses from the host bacteria by actually just putting them in a blender. So after giving them the chance to infect the host, they knew that whatever the virus inserted in the bacterium to transform it would be found in the bacterial cells. What they then did was centrifuge samples and looked at the radioactive material found in the supernatant, this fluid, and in the pellets. And what they had found is the pellet that contained the heavier bacteria was able to show that the phosphorus, the radioactive phosphorus, was what had been inserted into the host cell, and that the radioactive sulfur from the protein coats was found in the supernatant. So they knew the virus bodies were made of protein, and then the genetic information that was inserted into the host was DNA. So that provided sort of conclusive evidence that DNA is the hereditary information that gets passed between cells. Now, once that was settled, then the real race began for identifying the three-dimensional structure of DNA. Now, an individual that early on had a significant role in this process was Erwin Chargaff. He was a chemist, and what he did was study the DNA of several different species, and he was able to determine that in any particular species, the percentage of adenines and thymines were approximately equal, and the percentages of guanines and cytosines were also approximately equal. So each particular species had its own unique percentage of A's and T's and C's and G's. Now, this is certainly a significant part of the puzzle, but at the time, no one knew the significance of this particular finding. What we know today is that DNA is a polymer made of monomers. The monomer of DNA is what's called the nucleotide, and a particular nucleotide contains three parts. Okay? There is the phosphate group, the 5-carbon sugar, there's carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5, and then this nitrogenous base. So if you want to visualize it this way, you have the phosphate group, the 5-carbon sugar, okay, I guess that's 5, and then this nitrogenous base. So again, this is one particular nucleotide. And then what is done is these nucleotides are linked together to form the polymer of DNA. So again, that's what we know now, and that's what was the focus of such intense research back in the early 1950s. Now, Maurice Wilkins and Rosalind Franklin worked together to try and elucidate the three-dimensional structure of DNA. And Franklin had taken images of the DNA molecule here through a process, or imaging process called X-ray crystallography. So what she did was actually take a look at the DNA molecule. If you can imagine looking down the length of a DNA molecule, sort of like peering down a straw, here you see the what eventually came to be recognized as the bases of the DNA molecule. Now, using Franklin's image, Watson was able to calculate and deduce that the structure must be a helix. He was also able to figure out how far apart the individual pieces or components of the molecule were spaced. So he had a sense of the distance across this helix. Now, Wilkins also gave Watson and Crick access to unpublished findings of Franklin, in which Franklin explains that she believed the two strands of DNA were anti-parallel or ran in opposite directions, that the sugar phosphate backbones were on the outside of the molecule and the nitrogenous bases were on the interior of the molecule. Pretty significant findings. What they did was take Franklin's work with her images and her structural hypotheses, and they combined with that measurements related to Franklin's image and the combination of that and Chargaff's rule. Now what they found is that when they combined two purines with one another, like a and a G, then it was too wide for the image they'd found. When they added two pyrimidines together, it was too narrow. But when they had a purine and a pyrimidine, it matched the dimensions seen in the X-rays. So they knew that a purine, or A or G, a purine like A or G would match up with a pyrimidine like T or C. And doing that helped explain Chargaff's rule. So what they were able to figure out is that A would always pair with T and C always pairs with G. The other significant component of that is that when these bases are aligned in this way, they are able to hydrogen bond with one another or form these weak bonds between bases. So it helps explain the three-dimensional structure of the DNA molecule. Now at this point, what you should know, or what you should be familiar with, is Chargaff's rule, which is A pairing with T and C pairing with G. Just remember at center growth. You should know that DNA is a polymer made up of monomers. Those monomers are nucleotides. And that monomer is made of a phosphate group, this 5-carbon sugar, and one of two bases, either a purine or a pyrimidine. Let's see, we also need to be familiar with the fact that hydrogen bonds hold together those bases, and that sugars and phosphates form the rungs of, the backbone of the molecule, and that it's the nitrogenous bases that you'll find in the center.